Alright, today we'll be discussing what is long-term potentiation. And I thought this was a very interesting scientific concept because if you haven't really guessed by its name, it has something to do with the long term, and specifically it has to do with storing long-term memory. And with final exams coming up, we have to study and get the information into our long-term memory. So I thought, hey, why wouldn't it be interesting to discuss how that information is converted into the long-term memory on the molecular basis? So before we get into actually how this process generally works, we have to understand, well, where is memory stored? And it's actually stored in a place called the hippocampus. And the hippocampus is a group of curved structures, and it's located in the temporal lobe. And the temporal lobe is located on either side of our ears. And now that we understand this, we can understand how the process of long-term potentiation works. And this involves the communication of two neurons. And here I have the ending of one neuron over here and the beginning of the other neuron over here. And neurons are just the medium in which the brain and the nervous system uses to send electrical signals to each other so they can communicate with each other. And these neurons actually communicate through a space called the synapse, which is just a space between the two neurons. And actually the space, this thing over here, is actually called the presynaptic membrane because it's right before the synapse. So we call it presynaptic. And here, right after the synapse, this arrow over here, is the postsynaptic. Post meaning after, so postsynaptic after the synapse. So how do neurons actually communicate with each other? And we have these things called neurotransmitters over here. And neurotransmitters, they're chemical messengers which the neuron uses to send to the other neuron for a message to be sent. Well, the specific neurotransmitter used here is actually called glutamate. And glutamate is actually an excitatory neurotransmitter. And what an excitatory neurotransmitter does is, if you haven't really guessed by its name, it actually excites the other neurons. So it does that by opening up the ion channels over here and allowing positive ions to go in. And glutamate has to attach to two receptors, and they're receptors specific to glutamate, and they're called NMDA and AMPA. And these are just two names for the two receptors which glutamate has to attach to. And when glutamate attaches to this, it changes the structure of the receptor and allows for ions to actually travel in. So NMDA just reacts a little bit slowly, and it takes a lot of action potentials, which are just... And what an action potential is just basically what I'm describing, and how the neuron passes down information from this first neuron cell body to the next neuron cell body. So once glutamate attaches, you have ions called calcium and sodium actually rush in to these channels. And after that, basically what these ions do is make the negative membrane, this is already negative pre-existing, and once these positive ions come down, it makes this membrane over here more positive. And this actually in turn strengthens this synapse over here and allows for more electrically conductive ions to go through. So actually this calcium ion actually has more purposes besides acting with the sodium ion. It actually works as a second messenger, and what second messengers are they're just signaling molecules which basically set off chains of certain events. So it hits another molecule, and then that molecule just sets off a ton of events. And as the calcium ions go through, it just activates a kinase, and this is the example which I'm talking about as it's used as a second messenger. And this kinase, what it does is just it allows the synapse to, well, it allows for being more efficient at relaying nerve impulses. Well, now we just have a whole mess of things just happening, and so the neuron has to figure out how it could stabilize these changes. And as a calcium ion, which I mentioned before over here, it has to stabilize these changes, and it acts as a second messenger, as I said before, and this actually activates something called cyclic adenosine monophosphate, and this in turn activates other enzymes, which increases receptors, making it more sensitive to neurotransmitters. So then you'll have, and as a result, you have more ions coming in, and this repetitive stimulation of the receptors getting stimulated by the glutamate, and then in turn getting more calcium ions in, and then acting as a second messenger and activating kinase, and then cyclic adenosine monophosphate. This repetitive stimulation actually activates something called CREB. And what CREB is just like a binding protein in the nucleus of the neuron, which is this circle thing over here. And it switches genes on to direct protein synthesis. And the thing which it actually produces is called neurotrophins. And I'm just going to draw it in green. And what these neurotrophins do is they stimulate the growth of the synapse, which is all the way over there, and it also stimulates the structure elements of the synapse, and it stabilizes this entire flood and sensitivity. And this entire process is essentially how long-term potentiation is involved in long-term memories being stored. And this process at the end, which I described of how the synapse changes and restores back, is actually a process called synaptic plasticity. And synaptic plasticity is just, if you go into the name, plasticity meaning the ability to change, and synaptic meaning the synapse's ability to change is essential for long-term memory storage. And actually after that was a whole mess, and if you're following along, I think maybe it would have been easy to understand. Well, not easy, but kind of okay to understand. But I think maybe just looking at it brand new and looking at that diagram would probably been hard to comprehend. And I just realized there's a term which I forgot to cover, which is depolarization, and that's just, as I discussed before, as how the calcium ions travel into the cell, and it makes the membrane more positive because the membrane's already existing negative. So that's what depolarization is. And here we actually have the professional diagram here, which I got from a research paper. And yeah, and here are all the terms which we discussed. And if you want to use active recall to test what you have learned so far, then you should do so, and I highly recommend it. So I hope this was easy to understand, and I thought this was a really interesting scientific concept because it explained the notion of how long-term memory is actually consolidated on a molecular basis. And I thought maybe as people are studying, they could think of long-term potentiation as they're trying to get that information into their long-term memory. I hope this is interesting.